We are here with Andreas Wagner. He is Professor of Evolutionary Biology at the University of Zurich. Very interesting person. Before that he was at Yale and then at the Santa Fe Institute. Now he's at the University of Zurich. Welcome, Andreas, to our program.
Thanks for having me. Good afternoon.
So you are a professor of evolutionary biology. Is not every biology evolutionary biology?
So that's correct in a sense. I'm in the Institute for Evolutionary Biology and Environmental Studies, and to answer your question, what I would say is that there's two kinds of biologists, right? There is biologists that are mostly interested in how questions, right? How does the diversity of life around us, how is that organized, right? How do the molecules inside a cell interact to produce the cell or produce our organism, right? And then there's other kinds of biologists who ask mostly why questions, you know. Why are there so many different species, and why do molecules interact the way they interact? And I would argue that evolutionary biology deals mostly with the second class of questions. So not necessarily everybody is asking evolutionary questions, but evolutionary biology has a lot of the answers also for the how questions.
And specifically, what why question do you go after?
So most broadly speaking, the question that we ask is, why are organisms capable of producing novel features? So just to give you a little bit of background, if you look at the history of life, it's a history of almost 4 billion years, and you can look at it actually as a history of innovation, right? Of innovation in the sense that organisms bring forth new features and sometimes spectacular features. Think, for example, about the evolution of eyes. Right? The evolution of animals that can fly. When these features, wings and eyes, first were fully formed, they were surely transformative. They really catapulted their carrier into a completely new league of competition, if you will. Right? And these kinds of features are very visible and complex, and they're ultimately, of course, built on many microscopic changes, submicroscopic changes, and changes in the molecules that keep living things together and build them. And many of these molecular changes in and by themselves are also innovations. For example, if you think about the evolution of photosynthesis, right? The ability to actually extract energy from light and build living matter out of it completely transformed this planet, for example, the entire atmosphere, and filled it with oxygen. So we are asking fundamentally, how is it that organisms have this great ability to bring forth new things?
But these transformative big things like photosynthesis or sexual reproduction or eukaryotes, so simple bacteria to more complex types of cells. But there are few of these really big transformative events. Mostly, isn't evolution mostly micro innovation, little things here, little things there?
What I would say is that definitely that's the case. So these very complex and visible innovations are an accumulation of many, many small steps. But each of these small steps also brings forth new things, right? And in fact, sometimes individual small steps can also be in and by themselves innovations. Let me give you an example, right? So there is a substance that's called Pentachlorophenol. It was invented or discovered by humans. It's a fully synthetic substance. It has been used for a long time in the chemical industry as a solvent and for other purposes. It's highly toxic, so it's now highly regulated, so you can't just buy it in the store anymore. And it's also toxic to many organisms. But it turns out there's one organism that has arisen within the last 40 years or so. It's called Sphingomonas chlorophenolica. It's a bacterium, and it is actually able not only to live in the presence of this toxic substance, it is apparently also able to use it as food. Okay, so it feeds on a substance that would otherwise be highly toxic. And this organism carries an innovation. It carries a sequence of, it is able to perform a sequence of 4 chemical reactions that detoxify this substance and turn it into something that the cell can actually use. And for all we know, this innovation has arisen within a few decades since pentachlorophenol was first introduced by us. And it doesn't actually involve much molecular change. We know what that molecular change is, and it's not actually much. It's a small evolutionary change, the way you called it.
So it's kind of one little change or two little change in the whole DNA sequence.
It's a little more complicated than that. It turns out this organism seems to have imported genes from the outside that allow it to actually deal with this substance. But it's a small number of genes, and this kind of process where organisms import genes into their genome, it's also called horizontal gene transfer, occurs all over the world in all habitats, and especially among bacteria.
Yeah. Now, you do experiments, or do you just look at what's around us in the world and kind of go after the small innovations? Can you actually do innovation in your lab?
So we do actually both. We study diversity that's out there. That's very important, and we have huge amounts of information about that diversity, and it's mostly diversity in molecules and the things they do, because it turns out, of course, all our cells, our entire organism is populated and built by molecules, if you will, and molecules do all the work inside our bodies. So that's one thing we do. Another thing we do is that we actually study evolution in the laboratory.
And how do you do that?
So it turns out that you can, with a little bit of help, allow molecules to replicate in a test tube in very large numbers, in similar ways to how organisms would replicate. Of course, they're much simpler, but these molecules are much simpler, but they can also replicate. And we can allow these molecules to mutate. That is, change their composition one part at a time. You can do this with protein molecules and also with RNA molecules. So RNA molecules are very similar to DNA in their composition. They consist of, they are long strings of smaller building blocks. These strings are called nucleotides, and there's 4 different kinds of these nucleotides. So we have technologies to actually synthesize many of these molecules, allow them to mutate in the laboratory in a test tube, and apply selection to them for new functions.
So you'll actually have a fight. Let's say you take RNA molecules, you actually have a fight in there between the existing ones and then the mutated one, and then you see which one will replicate faster.
Right. So some of the things that molecules do is they catalyze chemical reactions. That is, they help chemical reactions proceed, right? And so we can select, there is some biochemical tricks that we use to do that. We can select molecules that are able to catalyze a reaction faster than other molecules, and those are the molecules that preferentially will survive in these experiments.
Sure.
And so we carry out multiple rounds of such mutation and selection. And just to give you an idea, I have brought you this little vial here. Not sure whether the audience would be able to see it, but it is one of those kinds of test tubes in which we carry out these experiments. And just to give you an idea, this test tube holds of the order of 1 milliliter. That is one. Maybe
you can turn it around so the camera sees it a little bit. Yeah, from forward. Yeah.
I'll just put it on its head so it'll be easier to see. Holds of the order of 1 to 1.5 milliliters, that is 1/1000th of a liter, and you could easily fit 10 billion, 100 billion molecules in there that all replicate and mutate, and that we can apply selection to. So this is actually a larger number of molecules than there is human beings on this planet, just to give you an idea of the dimensions.
But this is not, just to be clear, this is not life yet. This is just these are just molecules.
Exactly.
These are not cells that are competing against each other.
They're not self-replicating, for example. We have to help them replicate. So we use these very simple systems to study general principles that might hold for much more complex systems as well.
Now, somebody who is listening to us might ask, so this is all nice and fun for you guys, but does this bring any benefit to us, to society, to me as a consumer? Do you have an answer to that?
So, yes, I do. And first of all, you should understand that we do basic science, right? That is, we study fundamental principles of evolution. Right? We are not really working on applications, but you should be aware that there is a huge industry out there, biotechnology, that benefits from evolutionary processes in designing new molecules. For example, if you do your laundry, and you use laundry detergent, there is enzymes in there that help remove stains from your clothes. And these enzymes are engineered to actually be functional and be operational at high temperatures and still work at these high temperatures that they might encounter in your washer, right? If a diabetic injects insulin, it's not exactly the same kind of insulin that our body makes, but it's engineered in special ways to allow it to function better. And there's many, many other examples. And increasingly, this molecular engineering, or protein engineering as it's often called, uses evolutionary principles, actually uses exactly the same kind of approach to improve molecules.
Mm-hmm. So a pharma company will take this, let's say that laundry detergent company, ideally they would take this or a bunch of these and see which molecule will survive the best at that temperature with these number of stains, or
that's the idea,
or that concentration of stains. Okay.
Technically this can be very complicated and very laborious. You have to identify the right conditions under which you need to do that selection and so forth, but in principle that's the idea.
If I understand you correctly, one of your big ideas is that mutations happen, changes happen on the level of genes, but they don't really affect the outcome of a reaction chain, for example, or a building of a cell, or a building of an organism. There are mutations, but they don't really appear outwards. But it is important that these little mutations happen, because then once another mutation happens, you get a completely new level of innovation. Is that roughly what your big idea is?
Yes, I think that's one of the core principles that we and others study and have identified in different classes of systems, molecules, but also much larger classes of systems, for example, entire metabolisms. So to give you a little bit of background, okay, so I said proteins, for example, let's talk about proteins. Proteins are strings of amino acids. They're smaller molecular building blocks. There's 20 different kinds of amino acids, so there's many possible such strings. Now, if you take a protein that does something that's useful in your body, let's take an example. Sucrase. Okay, sucrase is an enzyme that is necessary for you to extract energy out of the common table sugar. That if you actually put sugar in your coffee and drink it in the morning, there's energy in that sugar, and you can only extract that energy if you have sucrase. And that sucrase is being squeezed into the gut by your body, essentially, to be released and to deal with the sugar molecule. It cleaves the sugar molecule into smaller pieces, right? So it turns out sucrase, like many other molecules, there's a particular amino acid string that it has in our body, but it turns out if you actually change that string in many different ways, that is by replacing one or the other amino acid, what this enzyme does not change very much.
Okay, it still works.
Exactly. And not only that, you could actually have long sequences of change. Okay? One change after another change after another change. It still does not change what the enzyme does. Okay? Another way of putting that is that enzymes like this and many others are robust to some extent to changes in this amino acid composition, to mutations, right? And so in other words, and so this process of changing amino acids is something that happens in nature all the time. And we know enzymes, for example, that have been around for many hundreds of millions of years. Okay? For example, globins, same kinds of molecules.
But why do we know that they have been around for 100 million years? We can't go back in time and see have they been around.
What we actually do is we isolate them from many different organisms, and we can show that, for example, closely related organisms like humans and chimps have enzymes with an amino acid sequence that is very, very similar.
Okay.
More distantly related organisms have enzymes with an amino acid sequence that is more dissimilar, and very distantly related organisms have a very dissimilar amino acid sequence of these enzymes, right? Yet the function of these enzymes has been preserved. Right? So we infer this relatedness by comparing more and more distantly related organisms. Right? So, and one thing that is remarkable is that for many enzymes, you could actually have two amino acid strings that produce enzymes with the same function, for example, the sucrase function that I talked about, but they have nothing else in common. Virtually all of their amino acids have changed. Okay. So if you actually showed you just these two, you would say there's no way that they could be related through a common ancestor, but in fact we know that they are. Okay? if you think of an enzyme, for example, as a molecule that is a solution to a problem life faces, for example, how to extract energy from a molecule, that's pretty remarkable because it says that there is many possible solutions to this problem, right? And that holds actually for most problems. This is one of the important things that we learned, okay? So there's, even if we think a problem is very difficult to solve, there is actually many different ways of solving it in nature, okay?
Do we know, let's take this little molecule sucrose that cuts the sugar in half, glucose and fructose, I think it is. How many different types of that molecule exist or are possible? Do we know that?
We don't know for this particular molecule.
Okay.
We know hundreds of them from existing organisms, right? But there's many organisms that we don't know anything about because they're long perished, right? And it's possible that there is forms of these molecules that have never been realized and would also do the same job equally well.
Okay.
For different classes of molecules, we can actually estimate the number of solutions, if you will, to a particular problem. And that number is absolutely astronomical. It's greater than the number of stars in the sky, okay? 10 to the 10, that is now a 1 with 10 zeros after it, is sort of a typical number that we'll find. And there's numbers that are much larger.
Wow.
Okay.
So for pretty much every biological problem, you have 10 to the 10 number of solutions, you could say.
I do not, biology is the science of exceptions to a rule, if you will. So to every rule that you find in biology, there is an exception. But I think the rule is that typical problems have many different solutions.
Yeah. What brought you into this field of research? What was the trigger point in your career where you said, this is it, I'm heavily interested in this, and I'm not going to go study birds or plants, but that's what I want to do? Well,
actually, that comes back actually to this distinction between how and why questions, right? And I think I remember a pivotal moment, although I didn't realize it at that time, was from in high school already. We had a very good biology teacher in high school, and this biology teacher explained to us the phenomenon of diploidy. Okay? Diploidy means that organisms like us have 2 copies of every gene. Okay? And I remember asking a teacher, so could that be because if something goes wrong with one of those genes, there's still the other one that does whatever it does and helps the organism survive. So a biology teacher told me, no, that can't be, okay? I accepted that answer at the time because, he was the teacher, I was the pupil, but I learned later on that, 10 years later, that people have done a lot of work on that question. And that actually, what I just said, that it may actually fulfill a fail-proofing function for organisms is not such a far-fetched idea. But this illustrates that I've actually always been interested in these why questions, and I would say that for most scientists that I know, they gravitate naturally towards certain kinds of problems. And from my perspective, those were these fundamental questions about why organisms are built the way they are, or why their genome looks a certain way, right? And much later, I became really interested in evolutionary biology, and there really, I think the most fundamental problem in evolution is how new things arise. Right? And I've always liked to work on very big problems. And that's how I ended up combining an affinity for why questions with interest in very fundamental problems, and you pretty much have the explanation on why we work on innovations.
Now, this idea of innovation for molecules or for cells, organisms, can we translate some of these concepts that you research into other fields? For example, innovation in business, innovation for products, innovation how we deal with each other, innovation of government forms. Is it possible to translate that in some way?
So for that, I'll again have to give you a little bit of background. Part of the reason why nature has been so successful in innovating is that it uses a small number of parts, like the amino acids of proteins, of which there are only 20 different types, like alphabets, like letters in our alphabet, and links them in a standardized way to produce many combinations of chains of such building blocks, right? Much like Lego does, with a simple number of building blocks to build many different toys and other devices, right? And so this is part of what makes nature really powerful in being an innovator, if you will. I would say these principles can translate to all the human areas of innovation that have the potential to use this combinatorics, that is, small number of elements, combine them in many different ways. And I think one such area, for example, one good candidate area is digital electronics, which is basically the basis of all our post-industrialized societies, right? And it basically uses computer chips. That's what it basically uses. That is Boolean logic chips, they're called technically. And what they do is they take a string of ones and zeros and convert them into another string of ones and zeros. So what they do is they compute Boolean functions. And they do so through an arrangement of simple switches on the chip that are wired together in a certain way to compute these functions. And in recent years, there has been a very exciting development in that area, that is the development of what is called programmable hardware. Okay? So those are computer chips that are not hardwired in the factory, but they can be wired on the fly by a user, or, for example, inside a robot that might explore its environment and learns. And so these kinds of chips are probably going to be very important in what are called autonomous systems. That is, robots, for example, that learn to navigate their environment, right? Okay. And so we actually analyze computational models of such chips, and what we learn is that there is some very similar organizational principles to those we find in nature. For example, there's many different chips that compute the same logic function, right? So I think once we start more systematically applying these kinds of principles to technologies like digital electronics, we may actually be able we will see that these principles are actually quite powerful and can be used to design what you might call innovable technologies prone or poised to innovate.
Now, looking forward 20 years, 30 years, your retirement, what would be your dream that you would want to have accomplished?
Well, clearly, it's very simple to state, to really understand how new things arise in nature, right? That's a very ambitious dream, and I'm pretty sure that I will not be able to achieve it fully. But I think it's much better to reach for the stars and to fail than to reach not very far at all. And I'm not sure who has said this, but I think it's a very good statement, that is much better to have some dreams not fulfilled than to have all of your dreams fulfilled in life, because then you have nothing else to dream about.
True. Good luck with the research.
Thank you very much.
Thanks, Andreas.
Thanks for having me.